to the Weekly Bioanalysis, a KCAS podcast.
Hello, and welcome to the fourth episode of the Weekly Bioanalysis. My name is John Perkins, and I'm a Senior Scientific Advisor at KCAS for all things related to Lcms technologies. I've been with KCAS for about 6 months. I have actually been within the bioanalytical industry for about 25 years, so I have a wealth of experience to pull from. I am here with my co-host, Dominic Guarino.
Hello, I'm Dominic Marino, the Senior Scientific Advisor at KCAS with a focus on LBA technologies and biomarkers. Like John, I have a wealth of experience in the bioanalytical business. Today's podcast, as always, is brought to you by KCAS. KCAS is a bioanalytical laboratory located in Kansas City, serving the pharmaceutical and biotech industries for over 40 years now. John and I are obviously the Senior Scientific Advisors at KCAS. If you have any questions related to today's podcast, we'd be happy to answer those questions. Our contact information can be seen at the bottom of this page.
We're very, very happy to have you listening again. As mentioned before, this will be our fourth week recording the podcast. So if you missed any previous episodes, they can be accessed at kcsbio.com and you can listen to the other episodes there. I have to say, I have a lot of fun working with Dominic on this and on the previous weeks. So now that we're 4 weeks into this podcast, what do you think of it so far?
Yeah, John, so far it's been really great. It's quite flattering to see all the letters that have come in from fans online, some praise here and there. Haven't had any bad remarks yet, so that's good. My family enjoys it. Certainly friends that have listened to it have liked it, and a lot of colleagues. But we're getting a lot of good feedback from the industry and really enjoy making the podcast.
As we've mentioned before, we started this whole thing because of social distancing due to the COVID-19 virus. Even today, Dominic and I are thousands of miles apart. He's in Kansas City, I'm in upstate New York, and our technical team are scattered around the Midwest. By starting this podcast, it gives us the chance to remain connected to you and our customers and our colleagues. It's an opportunity for you to get to know us, and even more, it's a chance for us to talk about what KCS can do to help you. As always, we'll focus on the latest news, then look at a topic of our choosing. If we get any feedback from any of you, we'd be happy to discuss that in a later podcast. Right now, I'm going to pass it back to Dom to talk through the agenda for this week's podcast.
Thanks, John. There's changing public perception of the pharma industry, and we'll go over what that is. There's also a conference called Workshops in Regulatory Bioanalytical, which is going virtual, and we'll touch on how that will play out. Additionally, we'll talk about new services and technology at KCS that we've brought on in the last, 6 months or the early part of 2020. And then we'll look at collaborative approaches for large molecule bioanalysis, meaning whether to look at a ligand binding assay platform or some sort of hybrid Lcms. So John, let's just dive right into the news and resources. What's the latest on COVID? There's been really a lot of sort of speculation out there. I know Fauci came out and said COVID didn't come from China. That one caught me a little bit off guard. I don't know where he's getting that from. I'm not saying it did come from China, but that seemed to be one of the biggest, most controversial things when it came to COVID this week. There was also, of course, some approvals. There continues to be a massive surge in pharmaceutical efforts towards a vaccine. Antibody testing still in the limelight. I saw recently that the state of New York wants to do where you're at, John. They want to test 150,000 people in the next month or two. So COVID still dominates the news, but those are kind of the few things that stuck out to me this week. How about you, John?
I haven't got a great deal to add. I think Fauci's comments were more based that the virus wasn't lab-based from China rather than it not coming from China, but I'll look into that further.
Sorry, I misspoke there. You got it, John. He came out and said it couldn't possibly have been lab-based, but I'm pretty sure that hasn't been determined yet. But it's interesting, and it's still something that'll play out. Of course, we're not a political show, but there's a tremendous amount of light being shined on the Chinese government and how they handled things.
I think there's light being shined on every government and how they're handling things, and maybe we do talk about that in the future. One thing that just came through this morning was there's actually someone doing a study in terms of delays on clinical trials. And the latest count reckons there's actually over 1,000 clinical studies being put on hold because of this. So it's having an effect on drug development. I think it's a case of things are being prioritized to move things forward. But our phase 3 studies are being impacted. We've certainly known of customers that we're talking to have put some of their trials on hold while they try and move their most promising things forward. So that was one thing that came through today. That's basically being followed by a company called Bioregnum, who are tracking the effect of COVID on clinical studies. But as we said at the top of the podcast, we're not really focusing on COVID this week. Let's see what else is out there in the news instead.
Sure. There's been quite a bit of activity around pharmaceutical mergers and acquisition. One that sticks out to me is a company called PTC Therapeutics acquired Sensa Pharma. And that was kind of a little known acquisition, however, one that I thought made some news.
Yeah, I think some of this plays into the economic effect of COVID on business in general. The last recession we went through was 2008, which is largely down to financial crash. And for those of us who've been part of the industry at that time, we really saw our pipelines just dry up completely as basically all the pharma companies halted research and almost waited out the recession as it happened. That was the effect it had on us. So this time around, although there's been an economic impact, we're seeing less effect on the pharmaceutical industry. Obviously, mentioned that some clinical trials are going on hold, but we are still seeing things moving forward. We are still seeing fresh opportunities, and actually, we're still seeing economic activity with pharma companies. So, for instance, we're seeing there's mergers happening or purchases happening. No big blockbuster purchases like we've seen in the past, but they are interesting. So one of the purchases that is quite eye catching is Alexion. Alexion have been in the news as one of the companies who are looking to develop drugs to address some of the conditions resulting from COVID-19. But they bought Portola, who have an approved product called Andexa, and Andexa is an interesting drug because it's actually there to prevent any life-threatening bleeding that may occur from any patients taking factor XA inhibitors like apixaban or rivaroxaban. So Alexion are building their portfolio and their footprint by actually buying an approved drug that to date hasn't penetrated the market like Portola had hoped, but hopefully with more muscle behind them, they can benefit more from this unique drug. Another interesting purchase was actually Menarini from Italy, bought Stemline, who are based in New York. Again, Stemline have an approved drug that Menarini can build on, but part of the philosophy behind Menarini's purchase in this case is actually to help give them a footprint on American soil. So it is interesting to look at the contrast between the business slowdown here versus the business slowdown 12 years ago, and see that things are happening. So it's not entirely optimistic, but it's not really a pessimistic viewpoint if you're in the life sciences industry either.
Yeah, John, I think some would have said that most investments have essentially stalled unless they were in the 11th hour. I think having these three sort of pop up within the last week have been somewhat exciting. I have heard that there are many deals that have been canceled or delayed. I think a lot of people are trying to figure out how much impact this will have on the near term and post Q2. Although if you're watching the news, you did see, unemployment's really high. Q2 is going to be challenging and continue to see some of that, but all the signs point towards improvement in Q3. And then subsequently into Q4, we should see a surge, and they're really predicting that in 2021, the market will bounce back.
We certainly hope so.
Yeah, I think one of the things that is falling out from this that's really impacting our business, meaning the CRO business, is, global operations impacted. People are really looking towards CROs, and a lot of signs are pointing towards not only will CROs be relied heavily upon in the next, say, quarter or two. I think a lot, even when things come back to normal, the CRO market is primed to continue to explode over the next year or two. And most notably, there's a lot of that work that's being done perhaps overseas that now the public perception of what was happening in China and so forth, you might see some of that returning back to the US.
Yeah, I think what we as a service company looked at right from the beginning was how quickly can we react to this and also not interrupt or show any break in our ability to support projects, and so that's been the skill of a CRO service provider, by being able to offer that uninterrupted service where our customers have actually been closed or had limited access to facilities, it really, hopefully, it will show the readiness of us and others in our industry to deal with a situation like this, and we'll probably get people thinking more, this is a good way to go if I do hit problems, that the CROs have shown that nimbleness to deal with the crisis, that they're worth working with in the longer term.
Yeah, these are tumultuous times for sure.
So moving on to the next topic, we did actually talk last week about drug approvals that had happened in April, and talked a bit about that. And the numbers that I was talking about were actually a little bit inflated. The link I picked up from the FDA site actually talked about drug approvals of new drugs, repurposed drugs, and vaccines and things like that. But when we actually look at drug approvals, we're really more focused on new entities. And also another thing that I said was the worst talked of a year where we actually had only 18 approvals. Actually, the worst approvals we've seen recently was 2016, where there were only 22 approvals over the course of the year. So last month, April, wasn't 9 approvals, it was actually 5, but still that's pretty good compared to 2016. And in terms of that, we're actually on course, or there's 17 drugs approved so far through May 6th. And so we thought it would actually be good, having touched on the approved drugs in April, let's have a quick discussion about what was approved January through March, and then we can revisit May approvals when we have our podcast in early June.
Yeah, John, I think one thing about the lower number in 2016, in part, I think was due to 2015 being a historic year. I believe it was well over 40 approvals that year. So I feel like sometimes you might have a little bit of cyclical nature to it when it comes to approvals.
It was actually 2014 was the historic one. So 2014 was 51, 2015 was 29, 2016 was the low point, and then we've actually picked up considerably since then. So 2017, 2018, 2019 have all been over 40 approvals, with the high in 2018 of 59. So 17 so far this year. A third of the way through the year, we're a bit short of 2018's pace, but we're well in amongst the pace of 2017 to 2019.
That'll be interesting to monitor that, John. I wonder what the impact of COVID will be on all of this. One might think it might actually fall off towards the end of the year, but certainly it's been pretty robust to date. As you mentioned, there are 17 approved drugs and 17 novel approved drugs, meaning not anything that was repurposed. This is the first time they've been approved. You did a nice little breakdown for us, right? Maybe you want to go over, I don't want to steal your thunder, John, but what was the breakdown of those 17 drugs?
I'm happy to share. Well, we'll just touch on the January through March. There's 11 of them, 17 to date. But again, we're still seeing a healthy number of small molecules, 8 of those, 3 antibody drugs. And it's a fair mix in terms of indications. There's 1 ocular, 3 oncology, 1 inflammation, 2 GI, two for pain. So there's been a lot of focus recently on migraine medications, and there was 1 of those. 1 cardiovascular, looking at the high cholesterol, and then 1 for Cushing syndrome, which is associated with high glucocorticoids. So up till now, we've seen a nice mix of things coming through, but the emphasis will slightly change going forward. We'll see what the impact is on future months.
John, I'm sort of looking at the additional 6, and 5 of them a small molecule. There's just the 1 large molecule. That's the ADC approved by Immunomedics. The other 5 are all small molecule drugs. So we continue to see that heavy small molecule approval, which is interesting. In looking at it, it does look like there's a fair amount of treatment for lung cancer. That still seems to be an area that people are focusing on, as well as breast cancer. And the breast cancer ones are usually in the triple negative. People that are they're in the metastatic state, meaning there are almost no other options for them. They've probably had surgery and radiation and been dosed with a whole bunch of chemotherapeutics. So these types of alternative drugs for late stage cancers are certainly very promising and really give patients quite a bit of hope. And then, as you mentioned, there are some others in the pain space, as well as things like multiple sclerosis. And then There's always the rare disease states. I see one here for Cushing's disease.
It is always fascinating to see this because you know that the things that we're working on that do get through to approval are either helping us, our family, or our friends, and it's it's that's why this is a really rewarding business to be part of.
It sure is. Makes it real easy to get up and work every day if you think you're helping save people's lives. Well, I think we got to move on to the main portion of our podcast, and we'll just jump right into our topics. The first one is the changing public perception of the pharma industry. I don't know how much you pay attention to industry perception, but the cable company is like the worst, right? Like they're the lowest. People hate the cable company. Next, right up there was pharma, but now with this recent surge in wanting a vaccine and antibody testing and everything related to COVID, the perception of pharma has increased upwards of 60%. I hope to see that continue.
I think it's probably helped with the fact that people want to get back to a normal way of life, and what's the best mechanism to do that? And it really is through what the pharmaceutical industry can do for them.
Yeah, I mean, it's shocking that it was ever so low. I mean, I think there's always this, oh, they're gouging for pricings are so high. And, some of these newer therapies, the CAR T cell space, the gene therapy space, as well as enzyme replacements, I mean, these things, they're the price tag for annual treatment can be over $100,000 a year. But, they're expensive to make. I mean, CAR T cells can take upwards of 30 days in culture. And that comes with a huge cost. There's certainly other factors. I mean, if you think of what is it, something like 1 out of 15 might move into approval or even late phase. I don't know the exact numbers, but companies have to take many shots on goal before they get approval, and R&D dollars have to be recouped. So all of this, and again, I'm in the field, so I'm going to be somewhat biased, but I think the public perception around pharma and biotech is certainly more favorable and justified.
I think a lot of it is down to price and people see it hitting their pocket. You're not helped by the fact that you do have the odd case we've had recently where people have taken a known characterized drug and then they bought it as a company and then vastly overinflated the price that they're then selling it on for. And so those are what get in the public perception. There's no realization or very little realization out beyond us knowing when we're moving into biological drugs, just the amount of effort that goes into producing those. And like you say, the complicated nature of manufacturing and scale up to deal with a patient population. I know we're talking, gene therapies are beyond the 100,000 a year. I think there's been a couple of being priced at over $1 million for a year, but they're a one-time treatment that should hopefully set someone on track to be cured for their life where there was nothing for them to turn to before. But it really is the background information of the cost of research and cost of manufacture of those type of therapies has to be recouped. And that's not an easy story necessarily to sell to someone, but that's really that's the reason for the cost of drugs. But I do think that's why a lot of the negative perception is out there.
Yeah, you're right. There was a big stretch a few years ago of several companies that were purchased and selling a generic drug for like, I don't know, $15 a pill or something. They jacked it up to like $100 or $200 a pill, and that's just pure greed. We don't need that in the industry, but it did really, set a lot of bad press on the pharmaceutical and biotech side of things. I was watching CBS Sunday Morning News. Do you know the story of polio? Jonas Salk, he got absolutely no money for the polio vaccine. And in fact, they showed some footage of him where he said, why would I want to benefit from this? This is the public's domain. So I thought that was an interesting tidbit around how when you think of historically one of the best stories around solving a problem with polio.
Yep. So he's a great counterbalance to Martin Shkreli, who is the classic example of someone who took on a drug that was traditionally very cheap and then massively raised the price to take advantage of that. So he got the manufacturing license for an antiparasitic drug where its original cost had been $13.50 a pill, and he raised it to $750 a pill. So he's another bad actor, like Elizabeth Holmes, like we were talking about last week. Unfortunately, there's a number of those out there, but you hope for more Jonas Salks because what he did with polio was just unbelievably great thing to have done, and the benefit to mankind is overwhelming based on his generosity.
As you know, he's a University of Pittsburgh guy, and I'm so proud to have graduated from the University of Pittsburgh School of Medicine and spent time in the Salk building there.
So let's move on to the next topic. And this is looking at how people are adapting to remoteness in terms of conferences. So for us in Bioanalytical, we have two big conferences during the year, one in the US and one in Europe. The one in the US, WRIB, which, Dom, you're going to have to tell me what that stands for because I've never actually been to it.
You bet. It's Workshops, Regulatory Issues, Bioanalytical.
Okay, great. So it was scheduled to take place in April in Phoenix, and like I say, it's actually for bioanalysis. It's the premier place for us to exchange ideas, meet our colleagues, just get word out there about KCAS. But it's an excellent forum for just staying on top of what's happening in bioanalytical. It should have happened in April. Then, as obviously things changed, there was the announcement that was being postponed with the idea that we would get an update as to what was going to happen. The organisation did a survey of people who'd already registered to say, how would you like to do this later in the year? And the consensus was to do this as a virtual conference. So it's going to be an interesting exercise. The usual conference would be over the course of the week. The virtual conference will be over the course of 2 weeks. And they're trying to accommodate scientists from different time zones. So, on one hand, it will be very easy to make sure that you visit all the presentations, but there's all the additional stuff of a conference that you expect in terms of face to face meetings with people. That's going to be the challenge. How do you have those pieces that you would normally be looking forward to? It's not just learning. There is the exchange of ideas, but a lot of those exchange of ideas happen outside of the conference setting where you're actually meeting up with colleagues. So it's going to be a very interesting exercise. But I'm interested in your thoughts on that, Dom. I mean, I always like the sort of mixing up, meeting with people in the evening and chatting, etc. That won't happen unless we have virtual like cocktail sessions afterwards. Maybe that'll happen. But yeah, what's your thought?
Yeah, that's you touched on the virtual cocktail. That is something they're going to try to do. I mean, I think it's great they're having it for one. I've been to the conference for the last 7 years. It's an amazing conference. You really can get a wealth of information from presentations, and obviously this week that one of the benefits is you will be able to see all the tracks. Before you kind of had to choose which track you wanted to see because they'd be going in parallel. There might be 2 or 3 different tracks happening. So that is nice that you'll be able to see everything. And I'm sure you'll be able to have some Q&As, and that's fine. But one of the huge advantages of having the actual conference and several thousand people being present is you get to mingle during happy hours, you get to mingle after someone gives a talk, you get to really engage speakers if you want to. There's a lot more dialect, a lot more ability to drill into questions. Like if there are 1,000 people online asking questions, I can't imagine that'll get answered. But those are the things that will really be a negative impact here. So there is some positive. You'll get to, again, hear all these brilliant speakers and see all this great information. But I guess I'll call it the camaraderie that goes on with a conference will be lost. And even some of the spontaneous questions that one has when engaging someone and going back and forth over conversation. And that's really where the juices get flowing and you really start to come up with good ideas. So I hope that's not lost here. I think it's great they're still doing something. And I do think there is a virtual happy hour, and I think there's like virtual meals or something where there's times when because that's another big part of the conference. The meals they usually have pretty good food. Of course, I always got to talk about food, right? So, but those things, they have these big, you get to have big, massive, there's like at least a 90-minute window where you can sit around and really mingle while eating. And that, I think they're going to try and do that online.
Yeah, and then we have a virtual exhibition to look forward to as well, so that should be interesting. I mean, I think it sounds like I'm being negative on this. I'm really not. I think not only have they, made it, they tried to make the timing so it's applicable to people in different continents, but they've also added new sessions and to also give more in terms of for people to contribute to. So yeah, I think it's great. It's going ahead. I think it'll be very interesting to do it for the first time and see how it all works out. In terms of talking to the two main conferences, the European equivalent of WRIB is the European Bioanalytical Forum. And I've never been to WRIB, but I've been to all but one of the EBF conferences. It happens in Barcelona every year and it's timed for November. So praying that everything is done so that we can get to Barcelona and not have to rely on virtual food and drink, because that's definitely not an opportunity you don't want to pass up on, is eating in Barcelona.
Oh yeah, John. I've been to Barcelona. I lived in Spain for 3 months as a high school student, and would love to return to Spain. What a great place that is. So let's move off of WRIB, or what's referred to as RIB, and we can move into our next agenda topic: new services and technologies at KCAS. John, why don't you start us off with one of our first and latest and greatest service offering?
So one of the things that we've introduced recently for our discovery service is a powder to report approach. So as a bioanalytical provider, we're a relatively small part of the drug development process, but a very important part of the drug development process. When it comes to discovery work, people really prefer to have that all-in-one service where the animal provider also can do the bioanalytical. But we consider ourselves bioanalytical experts. And so what we've done is combined with other in-life facilities in the Kansas City area to combine our bioanalytical expertise with their in-life expertise to provide a service where if a customer sends us a powder, we will then coordinate every step of the way to produce data or a report at the tail end of things. So we can do the design for the study. We can then hand off to the in-life facility to do the animal portion of it, who will then send us samples. With them being local, they can send it to us by courier so we can have it rapidly after the end of the study. We will then do our typical extraction analysis and then produce data. And we're aiming to do that as quickly as possible, but also in as trouble-free a mode as possible to our customer, and really take all the work out of their hands.
Yeah, that's exciting. They call it powder to report, and we're looking to turn it around in less than what, 3 or 4 days, right?
I think the week's more realistic because there is some lead time in terms of getting animals and getting them settled in place, ready to actually do the study with. So there's some logistic that we can't control, but a week is probably a realistic kind of timing for all that. Speed isn't the only factor here. Sometimes it's just the having the partner who will take it off your hands and run it all the way through, and you don't have to worry about what's going on.
John, if you ship the powder to us and get your data in a week, that is definitely still very rapid. And it's not just limited to the small molecule side of things. We are doing it on the biologic side, specifically for monoclonal antibodies. We can do something called a universal assay. And without going into too much detail, that's a very rapid way to assess drug candidates in the biologic space. So another platform that we brought on recently is an ultra-sensitive biomarker platform called the SMCX Pro, made by MilliporeSigma. SMCX Pro stands for single molecule counting, and this system uses it's a bead-based system, and it has very little fluidics to it. It's an amazing little instrument that we've been working on for about 6 months now, and we're excited to be, in the process of launching our first salvo of biomarkers. Most notably is a biomarker called alpha-synuclein, which is becoming quite an important biomarker in the neurodegenerative space. And we're partnering up with MilliporeSigma, and we'll be having a webinar on that here shortly. Pretty excited about that. Any thoughts on the SMCX Pro, John, other than it's ultra sensitive, meaning it can detect, picogram or sub-picogram levels?
No, I'm excited to see what the data it produces and how we can, apply it in the real world. Sensitivity is definitely one of our biggest ongoing challenges. We see the same in LCMS, which actually takes me on to the next technology we want to talk about, where we're talking about large molecule hybrid LCMS. This is a technology that we apply, which is very complementary to the ligand binding type approaches for large molecule. But sometimes we hear of assays where there might be selectivity issues, where actually LCMS might be the approach to help solve the problem. Typically, large molecules are beyond the range of the mass spectrometer. So, in the case of a protein, we're relying on breaking down that molecule to find a specific part of the sequence that we can quantitate and use that as a surrogate for the bigger molecule. We digest using trypsin or other enzymes. We've used formic acid, lysine. There's others can be useful alternatives, and each of those break the protein at different parts of the sequence. And what we're looking for is a peptide chain that is of a good length, where it's high enough to be beyond any low mass type interferences, but it's also not too big that it's beyond the range of the mass spectrometer. So we can actually model the digestion using various programs, and then we'll experimentally prove that the peptides we're looking to track actually are sensitive enough and are free of interference. For internal standards, we use extended versions of those peptides, so that goes through the digesting process, so you have something to track the analyte that you're looking for. And we also implemented things like immunoaffinity extraction for really specific cleanup. In terms of chromatography, again, it talks about the sensitivity. We're comfortable with both micro and nano LC. And again, this is not just in proteins, we're looking for sensitivity, but as we're seeing increasingly hybridized types of molecules in terms of what are being administered as drugs. When we see pegylated compounds, quite often that affects how sensitive you can get for those molecules. So we're looking at whatever strategy we can get to get as low a level as possible to be able to answer our customers' questions about those compounds when they're administered.
Yeah, John, this is an ever-growing area at KCAS. We brought it on almost 2 years ago now, but it's just exploded in the last 6 months. And what we're seeing more and more of is something called a double affinity. So you touched on how you can pull down the protein with one antibody. There's actually a way to design it to do a second enrichment that can increase the sensitivity to unprecedented levels. And we're so blessed to have expertise in this area. And it's something that will continue to grow. I think it leads nice into, what part of the reason that KCS is such a great CRO is that we have all of this under one roof, where it really does separate us from our competition, in that the teams are able to leverage all of our LBA experience, because that's basically what the affinity capture component of it is, with all of our mass spectrometry and Lcms teams. And so it's amazing to see them synergize. We're able to pivot real quickly. If one platform isn't working, we can move to the other one real rapidly. I mean, it's an amazing atmosphere at KCS when we take on projects that are really challenging. So typically it's tried 3 or 4 approaches and they're not working. And then they're looking to us to take a look at it by affinity capture. And again, it's one that we're just growing and really solving a lot of problems. Dawn Dudefield is the director of that group, and she's a 20-year expert in doing this. And she is, training a team of scientists, and we got a whole army that can do it now. And what a special platform that is.
Yeah, I think it's really easy when you've been associated with the technology for a long time. So with me, it's LCMS for 25 years. With you, it's ligand binding for 25 years. To be blinded to the virtues and advantage of the other technologies. So you think, if someone comes to us with a large molecule, I'm immediately thinking LCMS. But by having everyone under one roof and that ability to pull them into the room to say, we have this issue, how are we going to address it, and what's the best technology to give the best answer? That's great. It's really, really tremendously powerful. Having been in a situation where we had the same services but geographically very separated, I've really seen the benefit of having it all under one roof for this kind of discussion to move things forward.
Yeah, there's a lot of efficiencies that we can gain. I mean, we can work at them in parallel. Whatever antibodies being done in the affinity capture component of things can be looked at on even the SMCX Pro. We've got a couple case studies where we looked at trying to detect proteins in parallel, both on our SMCX Pro as well as by affinity capture followed by LCMS.
And actually, another thing here is that where you do have these hybrid type molecules where you've got a lot of different functionalities, knowing that either technology may be able to hit the customer's needs, it's actually really good to have them side by side where you can actually conduct a feasibility on both, that can then determine the path forward to for the longer term of a project. So we've said it multiple times. It is a very, very powerful approach.
Yeah, and I believe we even have a webinar out there that you can go look up. The next item in terms of new services, we've got a 19-color flow cytometer that was brought on about 6 months ago. We're going to be bringing on 21-color flow cytometry. Flow cytometry has been around a long, long time. It started back in the 80s. It was 4-color, and then it evolved to like 5, and then it got up to 8 to 10, and then it was at 10-color for Quite some time, but recently there's been some advances in technology that allow us to go up to 19 and now even up to 21 color. And what I mean by that is this is an incredibly powerful tool. It uses something called hydrodynamic focusing, which allows for single cells to pass through a laser. And as you pass through the laser, you label the surface of the cell with upwards of 21 different biomarkers. I mean, this is an incredible advancement to allow us to understand the cell surface. I've been studying cell surfaces for a long, long time. I'm still not sure if we know more proteins that we can detect than those that we can't, meaning it's a very dynamic structure. The cell surface is constantly treadmilling or constantly ebbing and flowing up and down. But this instrumentation allows you to delineate cells at an unprecedented level. And again, we're so lucky to have someone by the name of Dr. Joyce Slusser, who has been for upwards of 5 years now, and she has got upwards of 30 years of experience. She even wrote some of the software on that flow cytometer while she was working at Beckman Coulter.
So talking to someone who really knows very, very next to nothing about flow cytometry, what's the benefit of having the additional colors?
Yeah, that's great. It allows you to do a couple things. So if you had to run 20 colors in the past, you'd have to run a couple different tubes, and that required a tremendous amount of data manipulation. And it also prevents you from seeing all of them on the cell type at the same time. There's the real benefit. Now you can put 21 targets on the cell, and that allows you to look at different subpopulations of T cells, different subpopulations of even some of the tumor cells themselves. I mean, it just allows for a much more robust thing. And really, it is in that T cell space. Of course, there's B cells and NK cells. And then it helps with differential diagnosis of the patient. So, for instance, in the leukemia lymphoma space, there are over 300 different types of leukemia lymphoma. And flow cytometry has really paved the way to allowing a physician to tell what type of leukemia lymphoma it might be. So I hope that shines a little bit of a light on some of the power around it. It does a couple things. It can save on data manipulation, data time, and it also allows you to look at a whole bunch of markers at the same time. Any other thoughts on flow cytometry?
Nope. I think it's good to learn more, and I need to spend time with Joyce and get even more input from her.
So with that, we're going to turn to the section of the podcast where we look at feedback and questions from our listeners. John, why don't you kick us off?
Sure. First comment is from Judy. I just listened to the third podcast and I hope many listen to it. I live in Kansas City area on the Kansas side. I appreciate the conservative approach Kansas has taken with re-entry to socializing. I agree with both of you that we do not want to have another Spanish flu situation. It has been 100 years. We have to have learned something. I totally agree with you, Judy. And yes, I'm glad that I'm actually in New York, which is also one of the states taking a conservative approach to this. And I really hope that we do take the right. We are considerate and listen to the experts in terms of how we should be opening up the country.
The next comment comes from Adrian. It's awesome, the podcast. Love it, love it, love it. Well, thank you, Adrian. We have a lot of fun doing this. We really appreciate all the people who are listening and all the feedback, and that helps make it a lot easier to prepare and get ready for.
Next comment is from John. I love the podcast. It really separates KCS from the other CROs out there. Thank you, John. Actually, what I think separates KCS from the other CROs is the quality scientists that we have on board and the level of expertise that they bring to the table. But I do appreciate your feedback.
The last comment comes from Derek. Listening to these two gentlemen relay facts about our time is both informative and entertaining. I hope the next episode about non-COVID items continues the quality this podcast has already come to represent for me. Well, I hope we didn't let you down this week. I think each time we go into this, we're hoping that we can keep raising the bar higher and higher. I want to thank all of our listeners for that feedback. That brings us to the end of today's podcast. Next week, we're going to continue to take a little break from COVID. We're going to let it sort of breathe a little. I'm sure we'll mention it in the news. But next week, we're going to talk about the evolution of Lcms and the regulated pharmaceutical space. John, this is right in your wheelhouse.
Yeah, I think the fact that you had antibodies last week, I'm going to take the podcast next week and we'll see where it goes.
Yeah, well, it couldn't possibly be a podcast without me mentioning pizza. It's funny, this week the wife told me she doesn't want anything to do with pasta sauce or pasta. So I don't know what we're going to eat over the next week, John.
And I honestly, we've no idea. I'm just glad that we're just about at the weekend. This has been a busy week. We've been on a lot of calls. I'm glad we had found time for the podcast. And it's definitely nice to look forward to a couple of days break before we have another busy week next week. For everyone out there, we're obviously, we really appreciate you listening. We appreciate your feedback, and actually we'd be interested to hear if there's any particular topics that you'd like us to touch on. Anything that you could share would be very welcome. So thank you very much again for listening.
As you mentioned, we welcome any questions or feedback or topics to discuss. We welcome your input. I hope you enjoyed today's podcast. Have a great week.